Good day, I am Mariel Therese E. Idalia, and today I will be discussing the definition of Molisch test, its underlying principles, and procedural steps. Before I discuss the definition of Molisch test, let's read first the overview of Molisch test. So, Molisch test is named after the Austrian botanist Hans Molisch. This test is based on the dehydration carbohydrates by concentrated sulfuric acid. It can detect monosaccharides, disaccharides, and polysaccharides, as well as compounds that can be hydrolyzed that carbohydrates such as glycoproteins and glycolipids. So, let's dive in. What is Molisch test? It is based on the reaction between carbohydrates and Molisch reagent, A-naphtol in ethanol, indicated by the formation of a violet-purple ring. So, the principle behind this test relies on the ability of sulfuric acid to dehydrate carbohydrates, converting pentoses into furfural and hexoses into hydroxymethylfurfural. These products then undergo a condensation reaction with A-naphtol, resulting in the formation of a violet or purple colored complex. So, secondly, how does the Molisch test occur? First, when carbohydrates are mixed with concentrated sulfuric acid. Second, leading to the formation of furfural and hydroxymethylfurfural. So third, why is this experiment important? First, it identified the presence of carbohydrates in food, biological samples, and unknown solutions. Second, differentiate carbohydrates from other biomolecules such as proteins and lipids. So this test is widely used in biochemistry, food industries, and laboratory research as a preliminary test for carbohydrate detection. However, it is important to note that while the Molisch test can confirm the presence of carbohydrates, it does not differentiate between different types of carbohydrates. Additional tests such as Benedict's test for reducing sugars or iodine test for starch are required for more specific carbohydrate identification. So, in conclusion, the Molisch test is a simple yet effective method for detecting carbohydrates in biological and chemical samples. Its reliance on the dehydration of carbohydrates and the formation of a violet-colored complex with A-naphtol makes it a valuable tool in biochemical analysis. While it does not provide detailed information about the specific type of carbohydrate present, it serves as a general screening test, making it an essential part of carbohydrate detection in laboratories worldwide. Now, let's dive into the Molisch test experiment. But before that, I will give you a fact about carbohydrates. So, the word carbohydrate is formed from the words carbon and hydrogen. Carbohydrates are combinations of the chemical elements carbon and hydrogen plus oxygen. In the nature world, carbohydrates are the most common chemical compounds used for food. So before the experiment, of course, we need some materials required. First is Molisch reagent, glucose solution, concentrated sulfuric acid, test tubes, measuring cylinder, test tube holder, test tube stand, dropper, stirrer, pipette, and distilled water. Here are the procedures of the Molisch test. First, prepare the Molisch reagent by adding A-naphtol in 10% alcoholic solution. Second, take 2 ml of given sample solution in a clean test tube. Third, add 2 to 3 drops of Molisch reagent slowly. Fourth, now add concentrated sulfuric acid along the sides of the test tube. Fifth, the acid layer forms a layer at the bottom. Lastly, if there is a formation of violet ring, then the presence of carbohydrate is confirmed. So as for the chemical reaction for the test, disaccharides and polysaccharides are easily hydrolyzed to constituent monosaccharides under the strong conditions. They undergo dehydration and condensation with A-naphtol. So thank you for watching my video presentation. I hope you learned how to do the Molisch test experiment, and I hope you learned and gain knowledge about this test. Thank you!